all species. Question 6. Right, the mechanism of sex determination is not the same for all species. In most mammals, the sex of the organism is determined by its genotype. Describe how genetic control determines the phenotype of maleness in mammals. Okay, they're all in bold and everything. They're trying to make sure that you're not going off in a tangent for anything else. So basically, it has to be the presence of the SRY gene on the Y chromosome. I was kind of surprised. I went and looked at the mark scheme on this one, and they actually just required the presence of a gene on the Y chromosome. They don't actually require you to know it was SRY. Okay, in some reptile species, the sex of offspring is environmentally rather than genetically controlled. Describe how an environmental factor can influence the sex ratio of offspring in such species. Right, so this is reptile stuff. It is temperature controlled. So temperature alters the ratio of males to females. Okay, we're not looking for actual specifics of this, but it's in fact, sorry, being clear, it's the temperature of incubation. So it's not the actual temperature at which there was sexual reproduction, but the actual incubation of the eggs. Okay. I think I need to spin down for this one. Here we go. Diabetes insipidus is a condition characterised by the production of an excessive volume of dilute urine. A hereditary form of the disease in humans is the result of an X-linked mutation in the gene coding for the receptor for the hormone ADH. The mutated allele X-a is recessive to the normal allele, which is X-big. Explain why males are more likely to be affected by diabetes insipidus than females. Okay, so here is your male. Okay, that means that they have every single X chromosome that is in the non-homologous area of the Y will show up. Okay, you can, like, if this is your X chromosome, here is your Y chromosome. This whole section here, which is your non-homologous section, regardless of whether the gene that you're looking at has an allele that is recessive or dominant, the recessive will always show up in the males. And that's basically what you're trying to say. So the males will always show up the recessive because it's in the non-homologous part of the chromosome. Okay, explain why carrier females are usually not affected by diabetes insipidus even though they carry a mutated copy of the gene. Okay, this is worth 2 marks. You're going to have to give a little bit more than just the fact that they have this. Okay, so what we have, remember, for all females, so that they don't have a double load of the non-homologous section, as it were, because that would be too much, they have random X inactivation of an X chromosome in every cell. So every cell has one or other of the X chromosomes inactivated. But that also means that 50% of the cells have an active ADH receptor. So you need to say X inactivation will randomly shut off half of the recessive and half of the non-recessive, but that means that 50% of them still have the active, so therefore that's enough for the kidney to function. Okay, that's it. And one more, sorry. A man who is not affected by diabetes insipidus has a partner who is a carrier. State the proportion of their sons that would be predicted to have the condition. So we've got a man who is not affected by diabetes insipidus, so he must be this. I'm actually going to split it up just so I can put it straight into the punnet. Okay, and has a partner who is a carrier. So X little a, X big A. So we do a cross. So we've got X big A, X little a. So that is a daughter who is a carrier. X in activation, 50% still working, she'll be fine. We have a daughter who has both dominant forms, so not even a carrier. We have a son who has got the activate or the incorrect function, and we have one who does not. Okay, we're going to completely ignore the girls because it just says proportion of the sons. Okay, and then we're looking for how many of them are predicted to affect it. There we go, 1 and 2, so 50%. And that's the question now.